Today we are going to get started with human reproduction, or at least get introduced to basic anatomy of the male and female forms in order that we can get stuck right in when we're back in class together. We're going to start off with the male. And of course, as you can see here, we have a nice cross section that is, in dissection terms, a sagittal section through the male. And just to orient you, let's have a little look. This nice big juicy looking red blob there, that is the bladder. And this nice hollow tube coming down the backside, well, you probably guessed, but that is our rectum. And then you can see tendon and bone in here as well. That's the bottom of our spinal cord, our tailbone, or coccyx, etc., etc. All right, so let's have a look at those overarching male structures, and then we'll have a look at them one at a time. Of course, the big shebang there is the testes. That's where the sperm factory is located. The sac that it's encased in is the scrotum. The tube running from the testes, so here's our testes, and then sperm get made, blah, 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 and then they follow up this tube here. That's called the vas deferens, and that vas deferens loops around and opens into the urethra, which runs through the center of the penis. Okay, and we have some glands. So the glands are in blue. These are the seminal vesicles. That's the prostate. That's the one that gets all the noise, but all three sets are equally important to us. The last one there, little guy there, looks like a bulb, is the bubble urethral. Okay, let's have a look at these one at a time. Oh. It wouldn't be a Carrie PowerPoint slide if everything was animated correctly. We'll just start off with the testes, of course. This guy down here, nice egg shape there, looks like the thalamus in our brain image. The testes, upon maturation of the male, they descend through a canal down into the scrotum. So earlier on in development, they're actually located inside the body cavity, and then they will drop down as the young male matures. The scrotum itself, this guy down here, is the pouch of skin that holds both testes below the pelvic region, and the purpose of that is to externalize them, to keep them outside of the body. And why do we want them outside of the body where they can be easily damaged? Well, that is because the temperature of the scrotum is a few degrees cooler than body temperature. And if the body temperature is too high, then our sperm will die. As it says here, this is important as sperm will not develop properly or survive at the regular internal body temperature. Okay, so even though it's a big, dangerous, wide open world out here, we want our testes to be outside. Now that nice little tube that connects the testes to the urethra is the vas deferens. That's this little guy here. Whoop, up and around like that. The vas deferens really is just a tube. It's a conduit. It's how the sperm get from the factory, the testes, to the ejector module here, the the penis. So the tube, again, on the inside here is the urethra. The vas deferens is continuous with it. If you are going in for a vasectomy, then that's where we get the name from, vas deferens, and ectomy, to cut. Okay, so historically they used to cut it right in half there and seal off two ends of that tube. Now they usually use a laser and just kind of pinch it shut and build up some scar tissue in there, and that that is a more reversible procedure. Okay, so let's have a look. The last one there is the urethra, the tube inside the penis, urethra. Comme ça. The urethra, as it says, enclosed in the penis, at the back of it has what's called an ejaculatory duct, and the ejaculatory duct regulates the movement of sperm and associated fluids. So these guys here together are collectively called the semen. So during an ejaculation event, the semen is given priority access to the urethra, as opposed to the urine. So during an ejaculatory event, the bladder's pathway is pinched off shut, and that way, as the bottom note here that you've been seeing since the slide began, says the urethra can serve as a passageway for both semen and urine, but never at the same time. Okay, so that ejaculatory duct is responsible for controlling traffic, as it were. Okay, let's crack on. Let's have a look at these endocrine glands now. So the seminal vesicles are these little blue globules at the top here, located right behind the, or right around the bladder. So again, that's the bladder there. Now the vas deferens curls around and then interacts with both of these vesicles in such a way that they can inject their fluids into that tube, into that conduit, and allow it to pass into the urethra. So let's have a quick look at these secretions. Now these seminal vesicles provide fructose as a primary source of energy for the sperm cells. Those little guys are high activity people, and they're high mobility and so specialized that they can't actually make much of their own energy themselves. And so they do need a sugar source for them to swim. So they have fructose from the seminal vesicles to provide that. They also, seminal vesicles, the paired seminal vesicles, also provide prostaglandins. Now, prostaglandins are a chemical that once they interact with the female system, they're going to trigger rhythmic contractions, as it says here, rhythmic contractions of the smooth muscle. And essentially, it's like creating a wave within the female to help those little sperms surf their way up towards the egg. So prostaglandins, contractions of the female. So after the ejaculatory event, they're going to interact with the female tissue and cause that to occur. This one, all of our glands down here in the male have this, the alkaline buffer. The alkaline buffer is to protect the sperm against the evil acidity of the woman's vagina. Also, the inside of the urethra itself is a little bit inhabitable to sperm. And so this buffer basically helps the sperm to survive on its passage up towards the egg. So the seminal vesicles, these little guys up here, have lots of big jobs to do for it. Let's have a look at the prostate now. So there's those seminal vesicles, and underneath it, the big purple bulb is the prostate gland. Now, a traditional check for the health of the prostate gland is, as I'm sure some males know, is for the doctor to have an actual feel of it. So they are going to insert their finger up here into the rectum, and you can see based on that proximity how close it is, and they can feel for rigidity of that prostate gland, see if it's stiffening in any way, etc., etc. Okay, so what does the prostate gland do? Well, just like the other one, it has that alkaline buffer in it to help protect the sperm. And it has one extra little role here, and that is to release enzymes into the fluids. So when the sperm come out, it's going to be mixed with all of these other secretions, yada, 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 into our semen packet. And, excuse me, as I was saying, the enzymes from the prostate gland are released, and what their purpose is to help the sperm cells swim. And they do that by, as it says here, increasing the fluidity. So they're basically partially digesting the thick secretion, so that's a little easier for the sperm to swim through. Our last ones here, so just recap: seminal, prostate, and now these little bulbule guys in yellow here. There's two of them, one on each side, and these are called bulbourethral glands. So bulbo for the shape and urethral for the location. They're right at the top of the urethra junction here. And they're also called Cowper's gland for historical reasons. And again, what do? Alkaline mucus-rich fluids. Again, to help protect the sperm from the nasty inside urethra and even nastier big old inside of the woman's vagina. All right, so that's our basic anatomy here. Just one quick little point. We didn't really talk about the penis, so I'm just going to point out that all of this nice spongy tissue on the inside here, this is heavily vascularized, meaning it's fed with a lot of blood flow. And so during sexual excitement, blood flow to the area increases, and it actually fills up all of these little spongy spaces inside here, and that's what actually causes the erection. So it's literally filled with blood in order to allow for that semen load to be inserted. Okay, let's move on. So spermatogenesis, in case you can't read sideways like that, spermatogenesis, or generation of the sperm, occurs with some really cool words that hopefully sound seriously familiar to you. So this is what we call a precursor cell. This is a regular cell that's going to be differentiated ultimately into a sperm. So first of all, we undergo mitosis. So the precursor cell undergoes mitosis and creates what's called a primary spermatocyte. And then we undergo 2 sets of meiosis. Of course we do. Meiosis 1 and meiosis 2. Meiosis 1 makes our haploid with 2 copies, and then meiosis 2 makes our fully haploid, good old half the number of our DNA complement. But of course, this guy here, he's not going to swim so well. We need to turn him into this guy here through a process called differentiation. So all of this process happens in the testes and the associated epididymis. Oh, epididymis, there we go. Which we'll talk about in a second. And it is the process where we, first of all, make the spermatid, which is a baby sperm, and then we turn it into the differentiated, the one we all know, the one with the head and the long tail, like that swims away. Note that spermatogenesis occurs continuously starting at puberty. So for the rest of the male life, spermatogenesis occurs. So let's have a quick look inside the testis itself. So singular testis, plural testes with an E. And here we go, and if we cut through that testis, we get this beautiful spaghetti-like configuration. And so the spaghetti is where the sperm are actually made. We'll get to that in a second. So just coming back, this is the tube that ultimately leads off to the urethra, the vas deferens, and then wrapped around the outside of the testis. So this guy here, that's the epididymis. Oh, I think I put a U on the previous slide. Oh, it's early in the morning for Carrie. Okay, and then the spaghetti stuff itself is a seminiferous tubule. Seminiferous tubule, there we go. Let's have a look. So, what is that picture? Well, I forgot to tell you. What we're going to do is we're going to take a cross-section through the seminiferous tubule, which is, again, the sperm factory. And this is where the sperm are developed in here. So we're going to have a cross-section through there, and this is what it looks like. So, and you can see we have all sorts of different types of cell and all sorts of different types of growth. Now, spermatozoa, this is aka just sperm. Okay, so the spermatozoa is the zoological term for it. And then you can see we've got Sertoli cells, interstitial cells, capillaries, etc., etc. So we're going to talk about Sertoli cells. Those are basically support. Interstitial cells are testosterone factories. So that is where a lot of testosterone is produced. So let's have a quick look here. Each testis is filled with seminiferous tubules, and these are lined with sperm-producing cells called spermatogonia. So those are like our precursor cells we were talking about on the previous slide. That's here, and you can see them undergoing our meiosis now, ultimately to become our sperm. After about 9 or 10 weeks, the spermatocytes differentiate into sperm cells. So even though this is happening all the time, there is about a just over 2 month delay from precursor to final sperm. But fortunately, we're making it all the time, so there's a continuous production. Whoops, okay. Sperm cells mature in the epididymis, which is a tube attached to the outside of the testes. Within 4 days, the sperm cells develop flagella and begin swimming at this point, and this is an important one, that we have some fail-safes in place. At this point, defective sperm cells are destroyed by the immune system. So not all of them make it. Not all of them have a chance. That means that the sperm that do make it through that filtration process are going to be the healthiest they can possibly be for potential for fertilization of that egg. Okay, so precursor, maturation, they finish up in the epididymis, but essentially very nearly ready to go as sperm cells here in the inside of that seminiferous tubule. Then off they would swim up the tubule into the epididymis, where they are finished off, ripened, and then checked over. These Sertoli cells, as I was mentioning earlier, are support cells that help to feed these tiny little guys here. The precursor cells have lots of organelles, but the highly differentiated sperm cells do not. And so, as you can see, these little sperm cells are latched onto the Sertolis, who's providing them with lots of good food, etc., etc. Let's have a quick look at the structure of the sperm itself. It says the mature sperm cell only has a small amount of cytoplasm so that it can move better. So that's that highly differentiated state. Because of that high level of differentiation, reducing the cytoplasm actually means reducing the organelles as well. It has limited energy. The vast majority of the mitochondria that it does have, which are, energy producing organelles, are concentrated near the flagellum. So that's here on our little sperm, in order to provide it energy to swim. So these guys provide energy for swimming. That's the primary purpose of this guy, is to get the nucleus, which is our DNA payload, to the egg. The head of the sperm, so up here, this little moon-shaped bit here is called the acrosome, and it's filled with enzymes. The enzymes are what help the sperm to fertilize the egg by dissolving the outer coating of the egg cell. And once they dissolve away, then the sperm can wiggle in there, and the nucleus will then be ejected into the egg for fertilization. So really fairly simple guy here. He's got a head with an acrosome. He's got mid-bit with all of his mitochondria, and then the tail itself, which is our flagellum structure. Note that, one little thing here, we put all of this amount of energy and work into these guys, and they can only live for about 24 to 72 hours at body temperature. So once they're matured, they have 24 to 72 hours to survive and to be ejected or ejaculated into the female and hope for fertilization to occur. Okay, last little bit here we're just going to talk about is the hormonal control of the male. So how and when all of this happens. So we're going to see some old friends here. So way up in the head here, we of course have the hypothalamus and the pituitary. Like this. And what we're going to see is that we have, switching colors, we have a hypothalamic hormone, a releasing hormone, and that is GnRH, that interacts with the pituitary gland. Now this will have to be, this should be in red. Pretend this is red, people. This is the anterior pituitary, because the hypothalamus is simply releasing a stimulating hormone in this case. The pituitary gland, the anterior pituitary gland, is going to release its gonadotropin hormones. The gonadotrophic hormones are then going to travel through the circulatory system down to the testes, and these are LH and FSH, luteinizing hormone and follicle stimulating hormone. Follicle stimulating hormone stimulates the production of sperm cells, and luteinizing hormone stimulates testosterone production. And then, of course, in good old-fashioned endocrine form, Once we have high testosterone levels in our system, it's going to feed back and inhibit the hypothalamus. And so, again, just like nearly everything in our body, we have a negative feedback loop. Okay? So again, hypothalamus, gonadotropin releasing hormones to the anterior pituitary gland. The ANT-PIT releases luteinizing and follicle stimulating hormone, which travels through the circulatory system to the testes. Luteinizing hormone stimulates the interstitial cells in the testes, in specifically the spaghetti-like structures there, seminiferous tubules, to produce testosterone. Follicle stimulating hormone stimulates those sperm to get started. All right, that is your quick introduction to the male.